right. Welcome to week 7, Bio 1031, Human Physiology. This is Ian Coates. Again, I apologize for this. This is the new medicine where they call you, and if you, geez, I received a call there from a physician. Hey, we can book you in for an appointment for November of this year. What are you doing on Tuesday the 17th? November, I don't know. This is the new medicine, so you have to take appointments when they come. So I'm sure you guys are heartbroken that there's no class there this week, and I hope you guys are enjoying your time and reading week next week. So have some fun, get some sleep, and get caught up. Let's go, week 7. This is a continuation on from last week when we introduced muscle to nerve. You're going to see a lot of, not a lot, but you're going to see some slides there that are similar from last week, and I've updated them. So let's go and have some fun, shall we? We saw this there, just the whole idea, the thick and thin filaments. Okay. In this case here, it's represented in a cartoon style. There's tug of war. Okay, so here are your thick filaments there, your myosin, here are your thin filaments here. Okay, these are your actin. And you'll notice there that when those heads from the myosin heads, they come in contact there with the thin filaments there, we call this there a cross bridge. The length of a sarcomere is from Z line to Z line. You'll notice there your H lines there, your I bands. Notice those. There's from last week there, but very good. So likewise, the short length of a sarcomere may be held in position there by the continued effort of the myosin heads. Nothing too crazy there. Last week there we talked about how the energy source there for a muscle contraction is ATP, but can also be creatine phosphate. Okay, this one here is more of a storage. You know, ATP, those phosphates, they're like hot potatoes, and if we can't put one onto an ATP, then we'll put the phosphate onto a creatine molecule. Okay, and just continuing on here with this slide here, you'll see there that what we've got is here's your creatine phosphate or ATP. Generally there you eat a banana, and that energy there goes right on to adenosine diphosphate. There's the extra phosphate on there, changing it into ATP, adenosine triphosphate. Or if there isn't any ATP available there, we can throw that energy onto a creatine phosphate, which then can be added there to an ADP. Nothing too crazy. As you guys will remember there's the aerobic pathway and the anaerobic pathway. Aerobic pathway occurs when adequate oxygen is available there. Okay, tends to be a little bit slower. Okay, supplies energy though for a long time rather than a short time. Okay, the anaerobic pathway, this is the beginning of every single exercise we do. I'll show you guys that there in the next couple slides. But the anaerobic pathway tends to be very fast, providing energy there for the first minute or so. Okay, and that gradually gets replaced there by the aerobic pathway if the exercise continues on past a couple of minutes. May occur when low levels of oxygen are available. Production of oxygen debt. Now what this means is, and you think of a 400 meter or 800 meter or 1600 meter sprinter or whatever, that whole idea of panting, getting that oxygen into you. That's what we call oxygen debt. Okay, sometimes called excess post-exercise oxygen consumption. EPOC. How's that one? Results in the formation of lactic acid, which requires oxygen there to convert back into glucose. Okay, so nothing, that's all just review. You guys have seen this slide there before, and I like this one right here. It demonstrates there with this one as well. I love the mathematics. When you can apply mathematics there to human physiology, it just makes it that much better. And you'll see there at the start of every single activity, exercise, okay, everything starts anaerobically, okay? But then you'll see there that aerobic production increases as the anaerobic production decreases. Okay, good stuff. Yeah, we talked about slow, fast, and medium muscle fibers. Okay, and this is where we think of, and the best analogy there is, just think of chicken meat, right? You've got the white meat and the dark meat. Okay, dark meat is dark because myoglobin, right? There's blood in there. So basically what we've got here, slow oxidative, slow twitch, fast glycolytic, fast twitch, and fast oxidative glycolytic, which is intermediate, okay? We'll go into a little bit more detail on these there than we did there last week, okay? These are your slow twitch, these are your dark meat here, small diameter, many large mitochondria, okay? Mitochondria. That's where we need, that's what we need, and there's lots of it here. Appears red due to large amounts of myoglobin, also known as red fibers. Okay, thick myosin filaments, type 1. And there's a couple of different kinds of myosin there that I'll show you there, and it all depends on the fiber itself, right? Wait until you get into cardiac, and when you get into, very good. These thick myosin filaments here, type 1, they contract at a slow rate, as we know, allowing ATP production aerobic to keep pace, avoiding fatigue. Now that's very important. ATP production is kept in pace with muscle contraction. Now these slow muscles, these are the leg muscles. Think of, the heavy muscles there that never really get tired, okay? And that's why they need the mitochondria. That's why they need lots of myoglobin there, oxygen. The ATP production is kept in pace, so these muscles do not fatigue. Develop tension slowly, resist fatigue, capable of prolonged and sustained contraction associated with their posture and endurance activities. Chickens walk slowly on their legs all day. Okay, and we've seen these slides here there from last week. You'll see there that you have your fast twitch. There's your eye muscle, gastrocnemius, soleus. Okay, they're actually a combination of the two, which we'll get there. Okay, we're just moving along. These are your fast twitch that tend to move about 5 times faster. Okay, and in reference there to a chicken, these are the white meat. Okay, large diameter, very few mitochondria. Okay, and really no myoglobin at all. They appear white. This is the white meat, right? Now, they have faster thick myosin. This is type 2X here. Okay, it's more efficient sarcoplasmic reticulum and T-tubules there to deliver the calcium. But if you're going to have a fast twitch, if you're going to have fast moving muscle, the problem is you cannot make ATP fast enough. If you go back here to the slow muscles here, the ATP production doesn't outpace the muscle itself, right? The muscle's not outpacing, and they're both kept at the same rate. ATP is continually supplied there to the muscles, so therefore the muscles really don't fatigue, okay? But not so in this case here. The cost for rapid contraction, these fast twitch, is rapid depletion of ATP. This is where these muscles here, they cannot make enough ATP to maintain this fast muscle twitch. Okay, where's my coffee? Oh, it's upstairs. So that's the problem here. There's lots of glycogen, but low mitochondria numbers, okay. Anaerobic, understandably, because ATP can't keep, the pace of production of ATP cannot be kept. Okay, these muscles fire too fast, so therefore they fatigue very, very, very fast. Okay, contract and relax quickly, providing short surge of power. Just think like chicken breast meat. Chickens don't really fly. They flutter their wings. They go 10 feet in the air and they come right back down because they get tired very fast. Okay, providing short surge of power associated there with sprinting. Okay, muscle size increased by exercising fast fibers, eyes, and fingers. Nothing too crazy there. These are the intermediate. Now, why are these ones called intermediate? It's because they're kind of a combo of the two. And that is not a small I. That is a big I. And you'll see there that large amounts of myoglobin here. They do tend to appear red, okay? Moderately fast myosin fibers. These ones here are called type 2A. They generate ATP through aerobic respiration and glycolysis. An example here, the soleus there of the leg has a high proportion of slow oxidative fibers, around 88%. So it's kind of a combination of the two, okay? Now, these slow oxidative fibers there, that was these ones here, slow twitch, okay? So these intermediate muscles, they have kind of a combination of both, okay? Fast and slow. Moderately resistant to fatigue, associated with sports such as middle distance. And here's two examples there, the gastrocnemius and the soleus, which we've got right here. You'll see how they go. Okay. As we are warm-blooded creatures, we get a lot of our heat there from muscles. And how do we do that? If you just rub your hands together, okay, you create friction, and from friction you create heat, right? So, and this is the whole idea. Skeletal muscle contraction produces waste heat that can be used to maintain the set point body temperature. Okay, here we're talking about a negative feedback loop. The whole idea there that you know, this whole the whole idea of the hypothalamus checking the set point, and you'll see what goes on here. If there's a decrease in temperature, okay, this is detected, okay, by the sensory nerves, okay. The temperature is decreasing, okay, so this information there is sent to the hypothalamus, which then checks against what their temperature should be, okay, 37, and what it is the temperature now, you'll see in here, it's actually 36. So what happens is now this information there is sent back, the effector back to the muscle, okay, and induce shivering. We want to induce, okay, friction, movement there of the muscles there, which will increase the temperature. And this doesn't shut off, say, so the temperature increases. The temperature is increasing and increasing, and if it gets too high, it'll also get sent back. That information will get sent back to the hypothalamus, okay, which will then check the set point. And let's say the temperature here is 39 or 38, then it'll cool it back down again. So this is what we're talking about there. Nothing too crazy. You guys have seen this slide there before. It's been a little updated, okay, when we're talking about motor units here. The motor neuron, somatic body, is one of those several nerve cells there that enters a muscle organ together in a bundle called a motor nerve. One of these motor neurons plus the muscle fiber to which it attaches, this whole thing is called a motor unit. Okay, there's your neuron, there's your Schwann cells there, and you'll see what's going on here. You see those branches down that go right down to the muscle fibers. Some motor units there can consist of only a few muscle fibers. The smaller the number of fibers in a motor unit, the more precise. Okay, so here we're talking fingers. The larger the number of fibers in a motor unit, so the more fibers there that are touching more of the muscle, you're going to get more contraction. You're going to get a more powerful contraction. Okay, it's not very precise. It's more for the power. So think quadriceps, think legs. Okay, and the smaller number of fibers here we're talking eyes or fingers, right? Very precise movement. Okay. Myography. Now this is where everything there we've done there, the first 12 slides there was a little bit from last week there just to get your brains into muscle neuron thinking. Now we're on to some new stuff here, myography. This is the whole method of graphing the changing tension of a muscle as it contracts. So here we've got a muscle, a muscle meat, all right, and it's hanging on a hook. And this is what, could be rat, could be whatever. And they've hooked it up here and they've got an electrical stimulator, okay? There's your electric jolt and you just stick it right in there and you can fire and that thing will contract. And contraction, the very word, means that the muscle will shorten. So as it contracts, this bar will go up, all right? And you'll see what's going on here and we can record this, right? This classic type of my graph is called a chemograph, records muscle contraction as graphs showing changes in length. An isolated muscle moves the pen upward during contraction, and the weight pulls the muscle and pen downward as the muscle relaxes there. Okay. Electrical voltage stimulates a nerve impulse to stimulate the fibers in the muscle. Nothing too crazy there. Okay. So myography, that's how we measure muscle contraction. Now, just to be clear, we will be using the same technology there for Lab 4, but it'll all be on computer. Okay, it won't be, we won't be having meat hanging on a locker or meat hanging on a hook here and doing this kind of stuff, okay? Introducing here a muscle twitch. Okay, it's a quick jerk of a muscle that is generally that is produced as a result of a single brief threshold stimulus there. To get the muscle to contract, an electrical stimulus of sufficient intensity, and you guys will remember threshold stimulus, is applied there to the muscle. The twitch contracts. The twitch contraction there has 3 phases. Okay, these 3 phases here, we've got the latent period, we've got the contraction phase, and the relaxation phase. Okay, the latent phase here. Now, you'll see what's going on here. This is the whole idea of the muscle contracting, right? That's all. It's no different than here. No different at all, all right? But you'll see what's going on. This latent phase here, and there is no stimulation. Really. As soon as we start to get stimulation there, we're into another phase, the contraction phase. So before the contraction phase, the nerve impulse travels to the sarcoplasmic reticulum there to trigger the release of calcium. So that's what's going on in the latent phase. The contraction phase here, this is the actual binding of calcium to troponin, okay, and the sliding of the filaments. So that's what's going on here in the contraction. So here, in the latent phase, the calcium is released, right? You'll know that calcium there binds to troponin, which then flips the other protein there, tropomyosin, releasing the active site so that the thick filaments can bind onto the thin filament actin filaments there. So the contraction phase, this is where the binding of calcium to troponin actually occurs and the sliding of the filaments happen. There's your contraction, okay? The relaxation phase here, so because the sarcoplasmic reticulum has a greater affinity for calcium than troponin, the calcium is sucked up again by the sarcoplasmic reticulum there into these sacs. Okay, so sarcoplasmic reticulum has affinity, likes calcium. Troponin likes calcium. But sarcoplasmic reticulum has a greater affinity for calcium. So when the calcium is released, okay, it starts to immediately pump that calcium back into its sacs, okay? The calcium is removed from the troponin molecules, shutting down the contraction, stopping the sliding. So you see, that's what's happening here. Because the sarcoplasmic reticulum is scooping up that calcium, as soon as it released it, okay, so this is all at 0.1 second right here. We need 10 of these to make 1 second. So it's happening very quickly. The calcium is released, it binds, it starts to get reuptake there by the sarcoplasmic reticulum. Now, but if we keep calcium in the system, we don't remove calcium from the system, we will have a continual contraction. And we'll show you some of these there, okay? Welcome to it. It's called TREP, the staircase phenomenon. Gradual step-like increase in the strength of contraction seen in a series of twitch contractions that occur 1 second apart. Eventually, the muscle responds with a less forceful contraction and the relaxation phase becomes shorter. If the relaxation phase disappears completely, okay, a contraction occurs. So what does all that really mean? This. That's what we're talking about. Again, another myogram, warming up. Now, the whole idea here that, you know, whenever we're going to do exercise, more so for old guys like me, but whenever you're going to do some exercise, you need to stretch. You need to warm those muscles up. And like whenever you turn on an NBA game or a football game or whatever game, we see these people playing the sport. But they are all warmed up and stretched up. They're ready to go. And you'll see here what's going on, okay? In warm muscles there, calcium ions diffuse through the sarcoplasmic reticulum more efficiently, okay, than they do when they're cold muscles. So that's what's going on here. The Trepp phenomenon or staircase effect is a step-like increase in the force of contraction over the first few in a series. So you see what's going on here? Okay, there's your single twitch, all right? And what's going on, you'll see here that the muscle contraction gets stronger and stronger, eventually plateaus. Okay, it doesn't keep going, getting stronger forever. But after the muscle has been warmed up, okay, the same stimulation here, you see, same stimulation, here we get smaller contraction. Here, another one there, we get a little better. And then here, now we're up to max. And that's where you want to be. Okay, so that's why warming up, stretching your muscles, getting them going before you exercise. Well, besides the very fact that if you don't warm your muscles up, there's a good chance you could tear them or rip them or, do some damage. But even from a muscle contraction point of view, warming up, you're not getting 100%. Okay, it's all, we want that calcium to remain in there and stay in there. So in warm muscle fibers there, calcium diffuses through the sarcoplasmic reticulum more efficiently. Okay, and more actin-myosin reactions occur in warm muscles. Cold muscles, no. In time, repeated stimulation of a muscle lessens its excitability and contractility and may result in a muscle fatigue, a condition in which the muscle does not respond to a stronger stimuli. So this is over time, okay? A warm muscle fiber contracts more strongly than a cool fiber because calcium also accumulates in the sarcoplasm of muscles that has not had time to relax and remove calcium. Pump it back into the sarcoplasmic reticulum, right? So therefore, after the first few stimuli, muscle respond to successive stimuli there with maximal contraction. Thought that was interesting. Just a little bit of, what Mama always said when you were young. You gotta stretch before you exercise. Well, Mama's right. Don't mess with Mama. Tetanus contraction. Here we're talking about a multiple wave summation. Summation, you'll remember that word there. You remember back to the nerve, you remember the axon hillock. Remember there you could have, on the dendrite, you can get a couple of different stimuli, and then they are added or summed up together at the axon hillock there to produce an action potential. So what we've got here, tetanus contraction, multiple wave summation, produced when multiple twitch waves are added together to sustain muscle tension for a period of time. Okay, so, and just to be clear here, these previous ones there that we saw here, where did they go? There's A and B right from your textbook, and then we've got C and D. So, these all came from the same page in your textbook. If you don't have your textbook. If you do have your textbook, have a look. All right. So, tetanus contraction, multiple wave summation produced there when multiple twitch waves are added together. Okay. You can have incomplete tetanus or you can have complete tetanus. Incomplete occurs when a rapid succession of stimuli produced twitches there that seem to add together. Wave summation there to produce a rather sustained contraction. Very short periods of relaxation occur. Okay. Always remember, if calcium is continuously available, a contraction will be sustained, right? Okay. Very, very important. As opposed there to complete tetanus there, when a stimulation is such that the twitch waves fuse into a single sustained peak, the summation of twitches there together so the muscles cannot relax at all. Okay, so just different things there, and I thought that was interesting. The whole idea there, multiple wave summation there. You can add these up, and you see these ones here. You see how there's little bumps here, right? Because they are still pretty defined, but when you start getting into these little tiny ones, right, and the bumps become smaller and smaller until we get basically a smooth line here. Nice graphs. Nice graphs. Role of calcium in all of this. We know what calcium is doing there, physiologically. Okay. Please know that there for the test, from start to finish. And for the test that's coming up there, week 10, I know according to the stimulus, according to the syllabus there, it's on all weeks past week 5. So week 6, week 7, week 8, and week 9, right? For the test on week 10. But you're not going to get away from some of the things there that we covered on the first test. We're going to still be talking about neuron. We're not going to go into heavy, is this a Schwann cell? But we're still going to maintain that the neuron is involved in this whole process. So don't omit it there when you're studying there for test 2, okay? Role of calcium, you'll see what's going on here. Stimulus is applied, okay? A single sudden increase in calcium availability triggers the twitch contraction. So here's your stimulus. Calcium is released, okay, and goes and does its thing, and then the twitch comes after. And here we're talking milliseconds, right? Look at this one here, okay, multiple repeated stimuli maintaining a high level of calcium. If the calcium is maintained throughout, we will have a prolonged contraction, okay? You see here the calcium went up, just one, then there wasn't enough calcium in the system to maintain, so the calcium levels drop. They're reabsorbed there by the sarcoplasmic reticulum in the sacs there, and you get one twitch. But if the calcium is thrown out and maintained, okay, so the actin and myosin are still binding, you're still going to get a contraction. Okay, welcome to tetanus. Good. You've seen this slide there before. Remember muscle tonus, the whole idea there that our muscles are never, ever really asleep. Okay, and it's an evolutionary factor there that if a bear or a dinosaur just happens to want to eat us, like right now, our muscles can save us and get us out of immediate danger, okay? Muscles there with less tone, okay, we call them flaccid. Muscles there with more tone than normal, we call them spastic. And you'll see here that there is some cross-bridging involved in here. Okay, there always is for all of the muscles. Okay, they're always on to some extent. They have to be. There's a new concept here called graded strength potential. Is it difficult? No. Basically, it's all based on the premise there that skeletal muscles, they contract with varying degree of strength at different times. Why? Because skeletal muscles are doing different tasks all the time. Like right now, I know you can't see me, but I'm lifting a phone. And then right now, I put the phone down and now I'm lifting a book. You're doing different tasks, okay? And it's called what we call the graded strength principle. And the metabolic condition of individual fibers influence their capacity to generate force. If many fibers of a muscle are unable to maintain a high level of ATP and become fatigued, the entire muscle organ suffers some loss of its ability to generate maximal force of contraction. It makes sense. Your levels of ATP drop, well, you're not going to get a maximum contraction. Makes sense. Conversely, the greater number of fibers contracting, the stronger the contraction. Now, you guys remember there from last week that a muscle fiber is 100% or 0. A fiber will contract 100% or it will not contract at all. Now, what's the whole difference there between getting a strong muscle contraction and not? Well, it all depends on the number of motor units that have been activated. How many neurons are accessing this muscle and providing a surge? The more neurons that are providing more message to a muscle, you're going to get more of a contraction. The smaller number of motor neurons that are interacting with the muscle and providing a signal there to the muscle, the less fibers you're going to contract. Okay, so therefore your muscle fiber isn't going to, the whole muscle, the whole quadricep is not going to fully contract if you don't have the ATP available to it. Factors contributing to phenomena of graded strength here. There's 4 of them here. Metabolic condition of the individual fibers, what's going on with them biochemically, number of muscle fibers contracting simultaneously. The more muscle fibers you have, the stronger the contraction. The number of motor units recruited. How many motor units are involved in this muscle contraction? If there's only one, you're going to get a little bit of the muscle contracting. Okay? Intensity and frequency of the stimulation. Absolutely. Absolutely. Okay? And look, when you're talking frequency, okay, the higher the frequency will alter the contraction. Strength of contraction compared with the strength of the stimulus. So after the threshold stimulus is reached, a continued increase in stimulus strength produces a proportional increase in muscle strength until the maximal level of contraction strength is reached. Okay, so what we're talking about here is you can increase the strength of the stimulus, but eventually at some point in time, the muscle contraction is going to max out. That's all it means. Okay. The strength of the contraction, you'll see what's going on here. And it's just the whole idea of warming up. And once it's warmed up, it's ready to go. The strength of the stimulus here. Okay. And we're continually, these ones here are sub-threshold. Okay. So here we have basically the start of an action potential. There's threshold has been met, minus 59. These ones here, you're not getting anything. That's why you don't have anything here. Because we don't have a contraction. We have a contraction here. Okay? And look, we're increasing our contraction, and the strength of the the strength of the stimulus is increasing here, and you'll see that the strength of the contraction increases up to a maximum, and then that's it. And look, they're even increasing the stimulus here, but it's not representing the contraction because that's all you're going to get from this muscle. Okay? Interesting. This one here, I put this in here because I want you guys to take a minute or two, like you can pause the video and just have a look. And I want you guys to basically be able to visualize what's happening at the microscopic level. Graded strength principle, continue. Which are false? Which are true? Just make sense of this. Think about it and why. So the longer the muscle fiber, the more maximal the stretch. Now, does that make sense? Like the whole idea, a muscle will contract. So it comes together, it gets shorter. Now, what happens if the muscle is already short? Are you going to get a good contraction? Well, no. The muscle has got to have some length to it in order to contract, right? A shortened muscle sarcomeres are compressed, therefore the muscle cannot develop tension. That one there, I like it. That's true. If a sarcomere is already short, you're not going to get a good contraction because it's got no place to go. Strongest maximal contraction is possible only when the skeletal muscle has been stretched to its optimal length. I like it. That's a good one. Okay. An overstretched muscle, though, cannot develop much tension because the thick myofilaments are too far from the thin. So you can actually overstretch a muscle, okay? And if you overstretch it, that means you will never have cross-bridging. Your thick filaments there can't grab a hold of your thin, so they're not even touching. We'll show you this there next slide. And you'll see what's going on here. Like here we've got a crunched up muscle that's not going anywhere. This thing you're not going to have a good contraction. Well, have a look. You got no place to contract. It's already, there's no H zone already. So this is why warming up and stretching your muscles is a good thing. Here's overstretched, and you'll see what's going on in here. This is also not good. And you see that the thick and thin aren't even touching, so they got nothing to grab onto. You're getting bupkis there. You're getting nothing. All right? And this is the optimal length up here. You'll see what's going on. We got a nice H zone. There's your eyes. There's your Z to Z. And you'll see you do have room there for a contraction. And you're going to get a good contraction there. All right? So this is called the length-tension relationship. The maximal strength of muscle can develop and is directly related to the length of its fibers. Makes sense, right? A muscle could contract if it's got room to contract. Here, does not have room to contract. Here, they're not even touching, so you're not even going to get a contraction. Okay? What else we got here? Stretch reflex. We've talked about this there before a little bit. We're going to go a little deeper here. This is where the load imposed on a muscle influences the strength, okay, of a skeletal contraction. Now think about that. Here we're not really talking about the muscle. Here we're talking about what the load, the 5 kilo box. I'm carrying a 5 kilogram box. This is what we're talking about, the load. So the 5 kilogram box imposes a force on the muscle itself. And it's quite interesting what happens here. We'll show you there the next couple slides. But this stretch reflex, where the body tries to maintain a constancy of muscle length in response to an increased load. Maintains a relatively constant length as load is increased up to a maximum sustainable level. When the load becomes too heavy and thus threatens to cause injury to the muscle or skeleton, the body abandons this reflex and forces you to relax and drop the load. So that's why you're carrying a box and you're struggling and you drop it. This is because the body is making you. And let's have a look here. This is quite interesting. Have a look at this there, where the strength of a muscle organ can be matched to the load imposed on it there by this is actually a negative feedback loop involving the spinal cord. Okay, and we'll just move on here. You'll see here that start here and we're going to move over here and then we'll go over here. So basically you see this person here picks up a box. All right, let's say 5 kilogram box, whatever. You pick it up and you'll see there that the muscles lengthen. Your body isn't sure what 5 kilograms, so you pick it up there and you'll see there that, here I'll zoom in. You'll see there that you pick it up, but then you see your arm drops a bit, right? You don't really know, you're trying to find your happy zone in carrying this. Well, how do you find your happy zone of carrying this is, we'll just jump over here. The strength of a muscle organ can be matched to the load imposed on it by negative feedback involving the spinal cord. So what happens here is this 5 kilogram load that is now being imposed on your bicep and your forearm there, and you see your forearm drops a little bit there. This information is detected, okay? And here's the actual length of the muscle. Or sorry, this is what's going on because you've picked up this 5 kilogram box. And this is the stretch imposed on that muscle. Well, this information there is sent there to the spinal cord, which it's now the set point of where that muscle should be for that particular load. It should be here. So the information then is relayed back to the muscle, and it's now corrected. Okay? Just to repeat that, this 5 kilogram load imposes a force there, okay? Now, where should the arm be in accordance to that force? Well, the spinal cord is notified there that this is what's going on with the muscle currently, this is what it should be, and makes the adjustment. The information is integrated in the spinal cord and a correction signal is relayed through the motor neurons back to the same muscle, which increases tension and returns the set point muscle back to its original or its set point length. Okay, and that's what's going on here. Great little slide here, muscle spindles there, stretch detectors. I like that kind of stuff. Have a read of that in the textbook. It's a nice one. Okay. Strength of muscle contraction, factors there that influence the strength of a muscle contraction. These ones here are pretty straightforward. The strength of a muscle contraction is based on 4 things there. I mentioned these there before. Metabolic condition, fatigue, TREP, okay. Recruitment of motor neurons, number of fibers activated, initial length of the muscle fiber, length-tension relationship, and the amount of the load. Okay, these all play a part. Okay. The next couple slides here, we introduced isotonic and isometric contractions there last week. I'll just go into a little bit more depth here. Isotonic contraction is the same tension, different length. So here's Here you've got a, you're at the gym and you're exercising. Here you've got a baseball. You're lifting anything, okay? And that's what's going on here. The ball isn't changing mass. So this is a 2 kilogram ball here, a 2 kilogram ball here. So the tension will be the same, but because you're moving it, you're going to have different lengths. The muscle is going to contract and extend, but still maintain the same tension. Okay? Resistance is less than the tension. Muscle contracts, resulting in work being done. Lifting something and putting it on the table, that's work. Changing the length of the muscle fiber sarcomeres, example, lifting a pair of shoes, lifting anything, okay? Because the muscle is moving against its resistance, the energy of a contraction is used to pull on the thin myofilaments and thus change the length of the fiber sarcomere. Contraction in which the tone or tension in the muscle remains the same as the length of the muscle changes. And there's two kinds here. There's eccentric and concentric. Okay, concentric contraction of those where the muscle shortens as it contracts their bicep. Eccentric, okay, is where the muscle lengthens. Okay, you see what's going on here? Concentric, you're pulling it towards you, the bicep contracts. Eccentric is the reverse, where the muscle is extending, okay, while it's going down. So concentric, shortening of the muscle. Eccentric, lengthening of the muscle. Okay. Isometric, okay, these are the ones there that it's the same length changing tension. Like a flexed arm hang, where you're just hanging two hands there from a bar. Or here, you're just holding on to the bar. Okay? And this is where the muscle doesn't change length, but the tension can change. Okay? Contraction in which the muscle length remains the same while the muscle tension increases. No work done. Expended as heat. Muscles don't contract, they develop tension. Tension is less than resistance. Holding oneself against gravity, can't lift an object because it's too heavy. Okay, not too bad there. Here's just a little recap of both of those there side by side. Have a look at those. There should be some good questions on the test on that. Moving on, like everything there we've been talking about has pretty much been skeletal muscle. Now we're going to move on to cardiac muscle. Okay, we'll talk a little bit about smooth muscle, a little bit about some diseases, and that's it. Okay. Easy peasy, nice and short. Cardiac muscle fiber, unlike other muscle fibers, cardiac muscle fiber is typically branched and forms junctions called intercalated discs, okay, with adjacent cardiac muscle fibers. Like skeletal muscle fibers there, cardiac muscle contains sarcoplasmic reticula and T-tubules, although these structures are not as highly organized, okay. I'm going to go back here. There's your sarcomere. Okay, there's a lot of similarities there between cardiac and skeletal. The cardiac muscle, they are striated. Okay, involuntary muscle. Okay, it does resemble skeletal muscle, but there's branching here, and it all starts there with the heart itself, right? The heart is just a conduit. Okay, and every heartbeat is signaled there, a signal there coming from the SA node, the sinoatrial node, your pacemaker up in the right atrium, and that electrical conductivity shoots over there to the left atrium, okay, and then down the interventricular septum, and around there, the rest of the heart there, the Purkinje fibers. So basically, that's why this whole branching, okay, the heart is basically one tissue, okay, where the electrical conductivity just goes through the whole heart. It's made like that and it's branched like that. So each cardiac muscle contains parallel myofibrils forming continuous contractile band around the heart chambers, just as I was just talking about, okay? Continuous contractile bands all around the heart chamber that conducts a single impulse across a virtually continuous sarcolemma. Love it. So basically the heart is just one continuous bit of sarcolemma. Plasma membrane there for the purpose of conducting their electrical impulse. Contractions are rhythmic and continuous to provide the pumping action needed to maintain constant blood flow. Fibers form strong electrically coupled junctions there with other fibers, and you'll see the branching there. Okay, good stuff. The structure there, the cardiac muscle. Cardiac muscle does not run low on ATP, thank goodness, and does not experience fatigue. Okay, you remember there when we introduced there the three kinds of muscle fiber. Skeletal there is very strong, voluntary, but fatigues. Okay, smooth muscle is involuntary, not strong, doesn't fatigue. Cardiac muscle is kind of a combo of the both. Okay, involuntary, very strong, does not fatigue, right? The T-tubules are larger and form dyads, not triads, as in skeletal muscle there with a rather sparse sarcoplasmic reticulum. Cardiac muscles sustain each impulse longer, okay, than do skeletal muscle, so therefore impulses cannot rapidly continue. See, the heart muscle, it's got its impulse, it's got its contraction. The reason it's spread out, because we don't want any of that trep. We don't want any of that happening. We want one beat, then another beat, then another beat. We don't want anything interfering with one beat to another. Okay? Cardiac muscle sustains each impulse longer than skeletal muscle. Okay? And cannot produce rapid enough to produce a tetanus. Cardiac muscle is self-stimulating. Cardiac and skeletal muscle contractions compared. Okay, so here we have a brief nerve impulse triggers a brief twitch, contraction of skeletal muscle in blue. Okay, but a prolonged impulse in the heart tissue produces a rather slow, cumbersome, drawn-out contraction. Okay. Smooth muscle there. There's no they do not look like the either two of the skeletal or cardiac at all. They're their own. You'll see there the blood vessels, digestive tract, bronchial tubes. Okay, some sphincters there. That's where this is found. Involuntary, the ANS, autonomic nervous system. The cell structure, small cells there with tapered ends. They can function as a single unit or multi-celled. Okay, one nucleus there per cell. No striation whatsoever, very small and very slow contraction. Because, you know, when you're talking a peristalsis, it doesn't need to be a hurricane. The smooth muscle, there's two kinds there that we'll kind of talk about, okay? As we said there before, there's small tapered cells there that have a single nucleus. Again, calcium comes from outside the cell and binds to not troponin, but calmodulin. Okay, so that's a little different. Smooth muscle, there's no troponin, calmodulin there, which then triggers a contraction. No striations, the myofilaments are not organized into sarcomeres. T-tubules are not present in smooth muscle. Okay, I'm not really going to go into any type of heavy detail about smooth muscle, but they contract very differently. You see, there's no T-tubules at all. Okay, instead, there's a loosely organized sarcoplasmic reticulum. Okay, remember the T-tubule, you know, you have your sarcoplasmic reticulum and the T-tubules go down into the muscle, right? Well, you look at these guys here, there's really no place to go down into. So that's why they only have this loosely in place sarcoplasmic reticulum, okay? So what we've got here, there's thin bundles of myofilaments there that span the diameter of a relaxed fiber, okay? This one here shows there the surface of the cell is rather flat when the fiber is relaxed. But have a look at contraction. Now it's not really the same contraction. I'm not really going to go too heavy into it, but it's not kind of the same contraction. It's more of what we call a balling up. During contraction of smooth muscle, there's sliding of the myofilaments causes the fibers to shorten and they kind of ball up, as you can see right here. It's kind of odd, isn't it? Okay, the fibers become shorter and thicker and exhibit these dimples where the myofilaments bundles are pulling on the plasma membrane. You can see them right here. Very odd, very strange. Okay, interesting though. Two kinds here. We have single unit and multi-unit. Okay, the single unit here, most common. Forms a muscular layer in the walls of the digestive, urinary, reproductive tracts. Gap junctions join smooth muscle fibers into large continuous sheets. Exhibits autorhythmicity, okay, and produces peristalsis. So peristalsis is associated there with single unit visceral, okay. Multi-unit does not usually generate its own impulse, but rather responds to nervous impulse, okay. Composed of many independent single cells, can form thin sheets, okay. This one here, I thought it was interesting there, the erectae pillae muscle, okay, of the skin. They, if you just look at the arm, you look at the hairs on your arms there, every single hair there has a little muscle called your erectae pillae muscle associated with it. They are responsible for the phenomenon of goosebumps. The whole idea there where the hairs stand up on end, that's your muscle there that does that one. Interesting stuff. We're almost done. We're on the tail end here. Just a big picture here. Function of all three major muscle types is integral to the whole body. Relative constancy of the body's internal temperature is maintained there by the heat. Caused there by the muscles, the immune system helps to defend muscle tissue against infection and cancer, as it does for most cells. And the fibers there that make up muscle tissue then are truly members of a large interactive society of cells that make us us. Muscle injury, we've talked a little bit about this there, and I think a lot of it there makes sense. Muscle strains and sprains, severe strain there can result from massive tear. It's all just about warming these muscles up. If an injury occurs in the area of a joint or ligament is damaged, the injury may be called a sprain. You'll remember there that if you sprain your ankle, you've now stretched the ligaments, right? And ligaments aren't meant to be stretched. Ligaments are bone to bone. Any muscle inflammation, including that caused by a muscle strain, is called myositis. Muscle infections, okay, tetanus, okay, disordered tetanus, abnormal condition of the central nervous system, caused there by the bacterium Clostridium tetani, okay. Poliomyelitis there, once a tragically common disease. Polio, okay, is a viral infection of nerves that control skeletal muscle. Polio used to be gone, but over the course of the pandemic, one of the tools there the scientists used there to monitor the levels of COVID there was to go down into the sewer systems and take samples and do tests on entire cities. And the city of New York there, they declared that there was a polio issue. Was it last year or the year before? Because surprisingly, they were detecting polio in the sewer water. Okay, it's got to be coming from humans somewhere. And the last slide that we have here, you guys could have a look at this. Muscular dystrophy, myasthenia gravis, hernias there. I've never had a hernia. I have a couple buddies there have had hernias there. This is where the muscle, weakness of the abdominal muscle there can cause a hernia or protrusion, okay, of an abdominal organ through the abdominal wall. Very painful. This is what we've got. I apologize if we just kind of zing through this. Please feel free to stop and start the video. I guess we're done now. But you guys can go back and watch this again. And again, I'm sorry for this. I would have rather much have seen you guys in class. But my doctor's appointment there is in exactly an hour. So I got to scoot. Have a wonderful reading week. I will update you guys in a day or two there on your lab 3s. Okay. I'm looking at giving you guys at least 2 to 3 weeks. I'll update you guys there, probably 3 weeks there. So nothing will be due until week 8 when we come back. But I'll update you guys in the announcements. Have a great holiday, safe holiday, have some fun, get caught up, get some sleep, and we'll see you guys week 8. Take care, guys. Be well.